Now carbon dioxide, okay, notice we've only talked about water at this point. Remember, two main things go into the overall reaction of photosynthesis, water and carbon dioxide. Up until this point, we haven't even mentioned carbon dioxide. So carbon dioxide is essential in the Calvin cycle, okay? 3 phases of the Calvin cycle. So here is where you may need to stop and go through this a couple of times. And it showcases this. Your text showcases it. You can, Google images of this all you want. I mean, you're going to be able to find really great resources of it. But you have it here. 3 phases that go on in the Calvin cycle. So now again, remember, what have we made? We've made NADPH. That's going to be vital in this process. ATP is going to be necessary in this process. Again, why do I need energy for this? I am building up a molecule. Okay, you always need energy when you're going from smaller molecules to bigger ones. So this is a cyclic metabolic pathway. The 3 parts are carbon fixation. So Ribulose Bisphosphate Carboxylase, which is called RuBisCo, is a molecule that is where basically the CO2 enters and meets. Okay, it's a 5 carbon. You are going to understand this better if you just remember a simple thing. Follow the carbons. Remember conservation of mass. What goes in is what has to come out. If I'm going to create a 6 carbon molecule, okay, keeping in mind, keep track of your carbons. It will help you kind of make sense of all of this. At the end, if you're on the end of a cycle, so we are going to have kicked out at this point a sugar molecule, and we are left with RuBisCo, a 5 carbon atom. This 5 carbon atom is going to meet up with a single carbon, right, CO2, a single carbon dioxide. And this is going to create an intermediate that is really unstable, a 6 carbon intermediate. Okay, so anyway, phase 1, boom, done. Phase 2, reduction. We are going to reduce ATP and NADPH to help rearrange a RuBisCo molecule into 2 3 carbon sugars. So again, I have an intermediate 6 carbon molecule. Let's take 6 carbons, break them up into 2 3 carbon sugars called pGal. Okay, and that's how we refer to it as. So it is important, the ATP in this case helps to reduce, helps to fuel this. NADPH helps to reduce this process, and basically that's what's happening there. So we get a reduction reaction. Then we're going to have regeneration, because ATP is then going to be used to bring back RuBP from pGal, and in this process we're going to also be kicking out a sugar molecule in this. So here's where you have, again, that idea, keep track of your carbons. In order to get 1 pGal as a product of the Calvin cycle, 3 molecules of carbon dioxide are joined with 3 molecules of RuBisCo. This makes 6 molecules of pGal, one of which is what we use. The rest stick within the cycle. So the other 5 get reassembled and recreated. Okay, so let's talk a little bit more specifically about this. So as you can see, carbon dioxide comes in, joins up with RuBisCo. RuBisCo, you get these intermediates. Okay, we are going to break that RuBisCo and that 6 carbon intermediate, okay, into pGal. pGal is going to get then reconstituted back into RuBisCo in the process of shoving out a sugar molecule at that time. So this is how we get it. So pGal, dude, for the win, man. pGal is important for this because we're going to need 2 of them to help build our 1 carbon glucose molecule. Okay, so that's your building block. 2 of them equal the sugar. Many of them can be put together for a polysaccharide for storage as starches. So kind of paying attention to that is going to showcase this. So a little bit on the history side, since it's called the Calvin cycle, Melvin Calvin is who discovered it using carbon-14 to basically follow the carbons. And that's what he did. He radioactively labeled them to trace carbon throughout. His efforts received him the Nobel Prize in 1961 for understanding this process. Oftentimes we call it about the ways in which plants fix carbon, or carbon fixation, because of it. And at one point in time, it was known as the dark reactions. That is a complete, I guess, misconception, if you will. And that's because when you say, oh, light reactions, dark reactions. No. Light reactions happen because light's necessary. Light independent, or light, where the reactions in which light is not necessary happen there. So it's not because these reactions don't just happen in the dark, they happen throughout the day. So for Kelvin, our input is we need 3 carbon dioxide molecules, we need 9 ATPs, we need 6 NADPHs. As a result of that, we create 1 pGal, 9 ADPs, and 6 NADP pluses, which are then used in the light-dependent reactions. Whew. How you doing? Good, because we're going to do a pogo on this, and you are going to understand this stuff better. So there is some versatility in this. So this is where we're going to start to wind this down. There's some evolutionary quirks that go on. RuBisCo evolved due to the fact of low oxygen conditions to help to have a method of producing sugar in the absence of oxygen. And the active site of RuBisCo, high affinity for oxygen, which obviously is a problem because then we have photorespiration. And this metabolic pathway, when RuBisCo grabs oxygen instead, is kind of a problem because it's a dead end. You don't get any sugar. So we try to avoid that. What can we say? So, however, lots of times, this isn't a problem, but as long as the plants can keep its stomates open, we're not going to have photorespiration because we're going to expel the oxygen that's produced. Raise oxygen levels, then it becomes a problem. So we get good gas exchange normally. There's a couple of types of plants that we have. One is called C3. No adaptations for minimizing photorespiration. You're kind of screwed. If there is a defect that prevents oxygen from being released, the cell is going to photorespirate. They'll occur at the same time, they're exchanged, sugars get transported through vascular tissue, yay, 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 life is good. Cool, right? Okay, so here would be like what a C3 leaf looks like, just to give you a little background, right? So this kind of gives you a sense of what's going on and just sort of some tissue histology of the plant. However, in an environment where you constantly keep the stomates open, this could be like hot or extreme weather temperatures, this can lead to desiccation, okay? And that's not really good too because you're going to have huge amounts of water loss. Closed stomates, you're going to increase gas inside the cells, you're going to decrease CO2 coming in, you're going to increase the photorespiration because, again, go back to that RuBisCo having a really high affinity for oxygen. No sugar gets made. I bet you can see the outcome there. Not so good. So we have some adaptations. There are C4 level plants. C4 level plants have spatial separation. These are ones that can fixate carbon in the mesophyll cells and incorporate a 4-carbon organic molecule, which is malate, with another enzyme. This can then be transported and we can take the CO2 and be able to produce sugar. So it's taking this, and even in when oxygen concentrations remain low, the light reactions can occur in these mesophyll cells, basically. All right, so here's sort of what's going on in that. So it's just, again, another way to fixate carbon in mainly really extreme kind of plants. So you have plants that fixation occurs during the evening when it's not hot temperatures. Carbon dioxide gets stored as an organic acid, generally as carbonic, you get this sort of carbonic-y acid 4-carbon thing. And then during the day, the organic acid store is then used to supply into the Calvin cycle. So we can still make sugar. So it's just an environmental adaptation that's occurred with these plants that live in sort of extreme-y kind of environments. So consider everything you've ever known, right? We've been able to exist because of photoautotrophs. They are the foundation of our food chain, the foundation of the energy that comes into our planet. They are the production of the oxygen that supplies us with our ability to live. And we find that plants can be really useful, not just for producing oxygen, but as an energy source. So there's lots of things we're trying with plants and what they can do for us, and they're pretty important little buggers. So yeah, we gotta throw plants their due, and throw photoautotrophs their due, because they're really important buggers. So holy crow, you made it. So make sure these are the things that you need to be able to do. Take your time with it. This is not a one and done thing. We are going to talk about it in class. This is just your first introduction. This may be, you know, this in fact could be the 5th time you've watched this. So, but utilize your resources. Understand the process. Understand what goes in, what comes out. Text has great cyclic reactions that showcase this. So have a great night. Get ready to talk about it and get ready for some cell respiration at some point. Have a great night, you guys. Bye.